quiet. Um, so I have updated the schedule for the upcoming lectures for the final half of the course, so you can find that in the syllabus. I've also uploaded a multiple choice exam from a couple of years ago that gives you an idea of the types of questions that we'll be asking during the exam. I hope to be able to give you a final answer on the status of the exam by the end of the day. I'm in touch with the exam commission people and the roster people, and they had to sync things. And so now I hope to be able to tell you more by the end of the day. You can find this in the files menu on Canvas. You can just download, it's called Re-Exam 2019. You can go through a few of the questions, or all of them if you like. And so just to make one thing clear, a few of the questions were part of the exam material beforehand, and we've taken that out now. Those are designated by pink, so you don't have to worry if you don't know the answer to the pink questions. Yeah, and so otherwise these are, so certain questions you'll find a bit puzzling right now, but that's just because you haven't had the lecture yet that goes into this. So don't worry if you're like, what does this mean? You will learn what this means before the exam. I promise. Let me check. I need to find the chat. Here it is. So if you have any questions during my explanation today, for those of you on Zoom, you can ask through the chat or just start yelling. I'll probably hear it. Can't guarantee, but probably will. Yeah. So my plan today is to briefly, so this will be a short lecture, right? To briefly go through the project notebook that you can find through GitHub, right? That's the project backbone notebook. So it's a single notebook that contains all three of the projects that you choose one from. Yeah. But the topics of the three notebooks, or the three projects, sorry, are part of the exam material. So you'll be like near the end of the course, you will have to also read the literature attached to the other projects. So you are exposed to all of the topics. So let me first explain a bit about how this works. So I've already sent an announcement out just now that documents the process, but let me just reiterate that. You create a group. You can create any group from 2 to 4 people. I don't care with how many people you want to do this, but more than 4 becomes a bit unwieldy. And then also I think it becomes hard to just give more than that amount of people a single grade, right? That stops making sense at some point. I advocate that if you can, find people with different backgrounds to collaborate with, right? So best would be to at least have one person from psychology and one person from AI in any group, or at least, so for example, from AI, you can have a psychology background. So make sure that you blend a bit in that sense, because you'll have complementary expertise, and that will really help the development of writing the report of the project. Yeah. So once you've created a group, so you go to Canvas, and there's under the People tab, you can see groups, right? It's set up such that you can actually put yourself in any one of those groups. So decide which number you want together with the others in your group, enlist in that group, and then go to the Google form that's linked in the announcement, where you can then say which of the projects you want to do as a group. And because I don't want all of you to do exactly the same project, because that would be dull. I'm going to set it up, I have set it up such that you get to say what your first preference is, your second preference, and your third preference. And I will then distribute the groups over the projects after the weekend. Okay. So try to create the groups, if you haven't already, and discuss which project you want, like before end of the day Monday, and then end of the day Monday, I will then tell you which project you have. Yeah, sound like a reasonable thing to do? Great. I'm really looking forward to this. In terms of the project, just you can always send me emails if you have any questions about how to do the projects. Right, there's a bit of literature attached to each of the projects, but try to find your own also. Yeah, that's really bonus points if you manage to like really look around in the literature and find things that you think are nice extensions, for example, to the project. And so try to also blend a bit the amount of time and effort you spend in doing analyses and in also documenting what you've done. So it can be a bit of a challenge in that sense, right? So you really have to divide your attention, divide your time. But I always like this sort of thing, so I hope you do too. You cannot find, just a question here, Oleg cannot find any additional info regarding projects apart from their names. So on the form, there is no additional info apart from the names, but I'm going to tell you what the projects are about in this lecture. And you can find the notebook that contains these projects through the syllabus. It links through the syllabus, I do believe, or through the GitHub page. I forget, both, I do think. So this is one big notebook, and it contains a few different parts. So the way that I like to go through this is to have the notebook open like so. There's an introduction that tells you a bit about what the idea is. Then there's three sections for so there's actually like an introduction section. Let me just collapse that so that we get to and then there's a couple of different projects that I'll collapse now too. And then there's this report outline. So the idea is that for this project that you collaborate on, what you end up doing is you create a report in the end. And an outline for this report that you'll create is already created for you, right? It has the outline that you should sort of try to follow, right? You want to make a title, an abstract, introduction, methods, results, and discussion. Okay. So what you can do is mix text and code in this notebook for your report. And what this also allows you to do is just do a lot of analysis at the top and then just produce plots at the bottom for your report. And so what you end up handing in as a group in terms of the assignment that's attached to each of those groups, you hand in the notebook. And that's what we end up looking at for grading. And what we'll look at is first of all, what you produce in terms of the report, the figures that are in there and the text that's in there. But this ensures that we can also see how you created those results. Yeah, so we will also be able to check your code. It's easiest to keep running this in Google Colab. It allows you guys to collaborate on a single copy of it. And you don't have to install everything yourself, because if you have to, we are depending on a bunch of different downloads and software packages. So if you end up having to install those, I'm very willing to help you try to do that. But that can turn out to be a sort of quagmire type situation, right? You think that you go in and you vanquish your enemy in a day, and then you get stuck, and then you're in there for weeks, right? So let me go through the different projects one by one. And you'll notice like which of the ones you like best. What we've tried to do is really cover a bunch of different topics. So we had lectures on encoding models, on patterns, on bold responses, all of that sort of things. And the different projects try to highlight specific aspects of an fMRI analysis. And when they, so when you do them, you will be sort of focused on a specific part of the things that you've learned during the course. So what I'll try to do now is go through the different projects one by one, and then also highlight why you would choose each of these projects, what's nice about them and what's maybe less nice about them. And then you can base your decision on that, or you can base your decision on, you have these things called dice. You can just roll them and then decide, or let that decide. Okay, so here we go. So all of these projects, I have done an initial analysis, right? So you don't have to start everything from scratch. There is, and that's also why it's called backbone, right? There is already a backbone, and you can extend on this. Well, my goal here is to not have you spend too much time on the basics of getting the data, for example, and also you don't have to program too much, right? So you don't have to do much more programming than the type of programming that you've already been doing in either the previous notebooks or in the top of the notebook here, right? So this isn't meant to be a programming heavy type of exercise, but if you want it to be a programming heavy exercise, then, go for it. Yeah, so if you want to create your own little Python package to do all this, I don't care. Not only do I not care, I appreciate that sort of thing. It's fun. So previously, when you ran these notebooks, the only thing that you had to do is like import NumPy, right? And there was one nibabel install involved, perhaps, but that must have been about it. Yeah. So the only thing that you had to do was just say import NumPy and import matplotlib, and then you could start, doing analyses. There's a ghost here, right? Yeah. Okay. Then you could start doing things. But now we're going to work with actual data, right? In many of the cases before in the notebooks, what you've done is you've worked with only single voxels data. And we're now going to do that across the entire brain. Yeah? And so that means that we're going to need a bunch more data. Of course, we have to download more data. But not only that, we also have to be able to work with these data, for example, in terms of visualization, right? And that means that we need to download and install more tools. So let me first show you the types of things that we're installing and why it's so cool. So you can, in principle, run this on your own computer, because most of the commands that we run here are, you could run them on any computer that has Linux or Mac OS installed. Windows is a bit more difficult. You'd need to install Windows subsystem for Linux. So you add another layer of complexity there. So if that was abracadabra, what I just said, then don't try it. Yeah, just keep working in Colab and that will just work. So the different packages that we work with, this nibabel is for opening files, and you've already used it once before. We're going to also use nilearn, which is another package. NI stands for neuroimaging, and then learning. So that's a package that actually allows you to do very, very powerful pattern analyses on fMRI data, like in 2 lines of code. So if you're interested in that sort of thing, if you ever start doing an internship where that sort of thing is useful, then you should have a look at the help for this package, because it's beautiful. And that's really also something that I may not have emphasized enough, like this sort of new version or new way of doing neuroimaging research really centers on using this type of open source software. And we've really seen this beautiful development of these software packages becoming more and more professional over time. So nowadays there are these beautiful packages, and I really think that nilearn is an example of really a very professionally crafted piece of software. And similarly, we have this PyCortex, and that's what we'll be using for visualization. We use that a lot for any type of visualization and working with surfaces. Yeah? What's that? Yeah, maybe. Yeah, if it's bothering you, yeah. Are you good at martial arts? Yeah, that's kind of annoying, right? Yeah, I'm wearing these, so I'm not hearing anything. Okay, so what Google does for you, just as a background, right? It just runs a little containerized Linux install for you, and what this ends up doing is based on Ubuntu. So you have this apt thing that Ubuntu type Linux systems have that allows you to install software. That's what it does. It installs Inkscape, which is this program that you use to do image processing with, and that's what PyCortex uses in the background. Now, PyCortex is also the package that we use to create those nice web viewers that I've shown you during the course, right? We looked at those ocular dominance columns, so that PyCortex has really two types of things. One is types of functionality. One is plotting the way that I'll be showing it here, which is on a flat map, right? So you can, it's 2D and you can just look at it. And the other thing is that you can spawn like a web browser type interface, and then you can do 3D things. So that latter part we won't be focusing on too much, but we will be using the flat map stuff quite a lot. So for each of the different projects, you will see that we have a lot of this flat map stuff. So first, what we do here is after we install and import all these things, did you find them? It's not.
Maybe it's
From there?
but you could hear close.
Ah, it really is a ghost. Yeah, I mean, if it's in this floor, I don't know, but yeah. Shall we just continue and hope for the best? That's the story of my life. Okay, so once we've installed these things, we now have a computer somewhere at Google, right? That should be able to process things the way that we want them to, or it to. And then we have to download data. Now, what I've done is I have taken all these data that we're using for the different projects, and I've uploaded them to Figshare, which is an open data sharing website. So you can actually go there and find the data, and there'll be descriptions about what the data are and that sort of thing. So I recommend when you are, stuck with a project, then also go to Figshare and look a bit about at what the extra data, like the metadata, are for the data set that you're downloading. And that sort of thing is, of course, really, really simple to get because Python just allows you to say, request some URL from the internet, and you can download it, and then you can just run unzip in the terminal. And that then means that that computer that Google has for you not only has the software installed that you want to be using, but it also has all of the data that you would be using zipped, like unzipped, in the right location, and we know how to find them. Yeah? So that takes some time whenever you run it, but then you're there. So there's a bit of explanation about where the data files are and all that. This is getting annoying once you are starting to pay attention to it, right? So what we're going to do is use this PyCortex for visualization a lot, and you see it on the screen here. Some explanation. I always import it as CX because I use it a lot, right? Then you start using these shorthands. Otherwise, people don't. And what you can do is look at the docs for this package. Also, if you're interested in being able to do a bit more, you can do that. But the basics are outlined in this notebook. PyCortex actually contains, or we've already downloaded, one subject's worth of anatomical information. It's called HCP 9999. It's actually something I partly made. It's the average subject of the Human Connectome project's surface. So like a standard surface, but it's a non-standard surface because I made it. But all of the data that we'll be using are in this standard surface format. So oh wow. I don't know if you're all in Zoom, but there's a nice comment here. One parapsychological theory on poltergeist activities, such as the knocking, called rapping, is that it is the unconscious release of mental energy piled up. So perhaps someone in the room is experiencing a lot of stress. Anybody? Everyone. Not everyone, I do hope. In any case, yeah, hopefully that released some tension. I don't know. What we are looking at in the data for all of these projects is not these 3D anatomical images or 4D functional images that we used before, right? At the start of the course, you visualized some of these images. That was just to get things going. It's much more modern to go through an entire processing pipeline and then start working with data on surfaces, right? If we are interested in the cerebral cortex, we are interested essentially in what happens in the gray matter of the cerebral cortex. That gray matter is a sheet, right? It's crumpled up inside the skull, but it's a sheet. And then the best reference frame for looking at things that are formatted in this way, like the brain is in terms of surfaces. So that's why we're focused on surfaces now. And the added benefit of that is that we don't have to work with 4D data, which can be confusing, right? Which dimension is which? Is it XYZT or TXYZ? I have no idea. I always forget. I have to check the Y dot shape. It's annoying, right? And so for a surface, that's easy because you just have a two-dimensional data set, right? It's just a two-dimensional block of data. If you have time series, for example, it's just vertices, like locations on the surface, and time. Yeah? And then you have separate data that tell you where all of those points are on the surface. But if you're doing a functional analysis, in principle, you don't really care about where things are. You just care about what the time courses are. So that's the type of analysis that you'll be performing also. So what I hope this does is it makes your life a bit easier if you're going to do an analysis on data like this. But there's also, for example, a bunch of anatomical information present and on these surfaces, and you can actually get that by asking PyCortex for surface info. It has this function get surface info for some subject, and then you can ask a type. So in this case, it's curvature. You can also do sulcal depth. There's a few of these that give you an idea of where things are on this surface. Yeah. Now, where are things on this surface? Well, I've tried to give you a few handles on where things are on the surface, because I realize that when you at first look at these flat maps, right, you go like, okay, yeah, there's like two hemispheres. I get that because there's two separate patches, but that's about all I know, right? So let me first try to alleviate some of that confusion, if it exists. What you, let me just look at whether I can, I do think that I found what went wrong with the server. Someone told me at the, yeah, this one. So some browsers, I think for most of us, what the problem ended up being is if you enter a URL like this, so this is an aside, but maybe it helps. If you enter a URL like this that doesn't have the HTTP in front of it, many browsers will try to prepend HTTPS, which stands for secure HTTP, and it will break because I haven't installed all the necessary certificates on the server. So if you change that to not have the S in it, then it will work. It's also the case, it can be the case that you have actually gone to the Vue website, which then tells your browser to enforce this behavior. And then you need to go into incognito mode and take the S out. Yeah. So that's the problem. It's that your browser thinks that it needs to be very secure, whereas I am a really shitty IT person and I don't know how to do that, right? So take out the S and reload it in an incognito mode. That should help you. Yeah. So, but let's have a look at what this looks like. If we, what I can do is, so here it's very clear what the left and the right hemisphere are, right? It's left and right. That's definitely something that we've all learned in kindergarten, right? And so this is very easy to see what's left and what's right, and where's where. We have an intuition because we can look at someone's head and we sort of know where things are, right? And that sort of correspondence already starts to break when we inflate things, right? So now things aren't where they are in the head because we've started to inflate it, and we can inflate it even further. I do think if I do this, and that will really inflate things, right? So now left is still left and right is still right. But we're already looking at this surface-based type of representation. Now, what we end up doing, I'll show you only one hemisphere, and I'll show you what that does, and then I'll try to find another website that actually emphasizes this even more. So what we do is we cut the center of the brain, right? So we're now looking at the right hemisphere. It's been inflated. What we do to create these flat maps is that we make cuts here from let's say here to here, and then we make cuts from here to here, and from here to here, and from here to here. So we cut these parts of the surface, and we cut out the middle part here. Yeah. And so there's a hole there. And you can imagine that it's like if you have an orange, right? You take out the crown, and then you make cuts along the thing. What you can do is you can start to flatten it out, right? That's really what we're doing, but then in the computer. So we have Blender, this nice program that people use for animations and stuff, but we can use it to do this sort of stuff. And so what you're always looking at is, you can imagine that if this is an orange and you're taking out the crown here, and you have cuts that allow you to peel it like so, right? Then what you end up with is always looking at this side of the brain. So also for the other hemisphere, you're always looking at the lateral part of the brain, and you have these flaps that are sort of flapping out. Yeah. And the idea is that you always do so when you pivot them out, right? So you always do so like this. So you have a left hemisphere and a right hemisphere, yeah, and they're exploded like so. So what that means is that when we look at a flat map like the one that we have here, this you can imagine, right? You came from something like this. Where are we here, right? And now you're looking at something like this. And what we did is specifically cut at the middle and then exploded. And what that means, if we look at a flat map, it means that the back of the head is in the middle. Because we've just, we've split someone's head open through like this and then folded their heads out like so. Really don't want to think about what that would look like in real life. But so anything that you see in the middle is the back of the head, and that's where the visual system is. So most of these things that we're going to be looking at are visual system oriented. Some are not so much. And then things in the middle will be here, like on these sides of the brain, or really on the sides of the brain. And anything that was in the middle of the brain, the medial wall, as we call it, is now going to be at the fringes of these maps, right? And also anything that's in the front of the brain, so frontal areas of the brain, will be at the furthest left and furthest right part of the flat map. Yeah? Does that give you a bit of an idea of where things are? Great. And we can, so you can actually go to this website here, I do think, which gives you an example of what this looks like. I recommend you go to this website because it's a really nice example of this encoding model idea that I talked to you about. Here, people were listening to stories, and they mapped out the semantic receptive fields of all of the voxels in the brain. This one has been cut. So let me show you the what happens when you cut a surface. So this is just inflated, right? And I'm gonna flatten it out. So now the cuts will appear, and it will go smoothly from this representation to a flattened representation. So pay attention. Oh, this always happens, right? Now I have to do it by hand. So you can go to the surface thing and then unfold it further. And what you see is that I just pressed on to the right, and you start to see these cuts appear. See that? There they are, right? And the middle just disappeared, right? So that cutting operation has now been performed. And now we're going to take that further and unfold it to its maximal flatness. Ouch, right? But that's what that does. I definitely recommend you follow this tour that they've created in this interface. So this is what you can do with this PyCortex, right? So this is just like the flip side of the PyCortex functionality. That thing about the poltergeist really worked, right? Someone was really relieved that this came out in the open, and then they stopped. Great. So that's what that looks like. Any questions? You can use this website. So it's directly linked to from the notebook. So you can use this website to gain some more understanding of where things are. And additionally, we've also tried to give you a few landmarks in this brain that tell you where specific regions of the brain are. Because we're so sensory biased as a lab, what we ended up doing was delineating the first 3 visual regions of the brain. We also ended up delineating motor and somatosensory cortex in the brain, which is on the central sulcus in the middle. I'll talk about that more in the neuroscience lecture. But there's some regions there that you can then easily find, right? They're always going to be drawn on the surface in lines. And then there's auditory cortex, which is here, which is like also on the sides, but lower. Yeah? So you can use those lines, those regions of interest delineations to orient yourself, like, where am I? Yeah. And you can try to, like, for example, just try to plot thickness of the cortex, for example, in this thing. You'll see that this thing that I talked about for the parcellations, the thickness, you actually see it. You can actually start to look at it. Right, so here this outlines a bit of where the cuts are, and it gives you an idea of how these things are related directly. Right, this comes from that Human Connectome project first paper. Then there's a part here that loads your data. So for the 3 different projects, there's different data sets. Yeah? What that means is that when you decide to run one of your projects, you need to uncomment some code here and comment others, right? So that you only load the data that is useful for your project. They're ordered exactly according to the projects in the notebook. Yeah. So in this case, I think the last time I worked on this, I was working on the hemodynamic response function mapper, and so I loaded it like this. Okay. So these have all been downloaded at this stage. Now, once you've downloaded the data, these data are in different formats. So they're all in the same format. Once you've downloaded and loaded them, they're 2-dimensional data. But they have gone through different stages of pre-processing for the different projects. Right? So be aware of this. One of the challenges of doing a project is to be aware of the status of the data, right? And this is done on purpose. You always need to find out, like, what are your data, right? And so that's one of the first things that you do when you start up your project, like, what are we looking at? And so what you can do is open the data and then start plotting it. So here, what I'm doing is I have this T-series raw variable because my data was just loaded. Yeah. And I can average that across the first axis, or well, in this case, the second axis, right? And that is the average of the entire time series over time. Yeah. So that gives me a single value per vertex on the surface. And then of course, I can plot this the way that I just plotted the curvature of the surface itself. Now I can plot the average anatomical image of the data. And you see that, for example, some regions, they weren't really recorded. They sort of dropped out. And if I can point to these, you can see that this is somewhere on the inside of the frontal lobe. This is somewhere at the bottom of the temporal lobe, right? So that was folded from the bottom of the brain outward. And this is the bottom of the frontal lobe, what you see here. Now, what are those regions? Well, those are regions where we don't have signal. It's because there's an air tissue boundary there, and we get really, really crappy anatomical images there in functional imaging. So we don't have any results there, and you can immediately see it from a scan like this. From a simple, so you're already analyzing data in this case, right? You can also look at the standard deviation over time. So what happened in these different regions, and it'll be something like this. Yeah. How does the brain fluctuate? Right? So it's the it's not the mean, but it's the standard deviation over time. But of course, you can also just start taking random vertices from that surface and then looking at the time courses, right? So this is just to demonstrate that there are these two dimensions in the data. If you do something where you concat or where you collapse over time, you end up with vertices that you can look at on the flat map, right? And that's always, that single line of code gives you a map of the brain, and you can look at it, yeah? So once you're busy doing any analysis, you can always go back there with a single line of code and have an idea of what you're looking at. That's the idea, yeah? And the other axis is time in the raw data. And that means that you can actually look at the time courses the way that you've been plotting time courses throughout all of the other notebooks also. Yeah, and this is just randomly choosing a bunch of vertices on the surface. But there's also other ways in which we can take specific vertices, right? This is just an aside. So yeah, this just outlines your that you do have to look at what type of data you're looking at, right? I'm not going to read the entire notebook, that'd be boring. What you can do though, and what we've added to the data that you download is an atlas, right? And we talked about this parcellation of how to create a parcellation. Now, we're not going to make our own parcellation, we're just going to take one, an atlas. That's based on the average of something like 1,000 subjects. And in that atlas, there are specific regions, right? So what that means is the atlas is just another data file. Yeah. Who's feeling really stressed? Sorry. So when you load this atlas, it's similar to a single time point functional data file. It's just vertices by some value. Yeah? That value is an integer value. It's a whole number. And every vertex inside some region has the same number assigned to it. Right? And in this case, there are 180 values in the atlas that we're using. There are 180 different regions in the brain. And so we can just use some value and select that value and see, so the value of 1 is in this first visual region of V1. But there are many more in there, and you can try out by just changing that value. What this does is it allows you to create a mask and only select voxels or vertices from V1, right? So you can test where is where does this have the value of 1, and then select all of the vertices from only V1. So you don't have to do your analyses during this project when you're doing things. You don't have to do the analyses on the entire brain. You can also do them on the single voxel by taking a certain vertex, or voxel or vertex. I'm just using them interchangeably. On the surface, you can take a single one, or you can sub-select a specific region also. Any questions about this? Great. So what you can do is also plot time courses for some region of interest, like an image, and then you can really see, like, is there anything structured happening over time, that sort of thing. Yeah. So this is just to highlight there are different ways of looking at the data, and if you wield these, you will be good. You will be able to understand what you are looking at. Yeah. So that's the basics for how to work with the projects. I want to propose that we have a little break now, and then I'll go through the different projects in sequence. Yeah. Why does this always take so much longer than I intended to? So here we are. I'm gonna start again. I hope that each of the projects in this notebook has been explained already, like in terms of text. So you can go back to this So I hope that I can keep it a bit brief. But then again, know thyself, right? I probably won't be able to. So the first project is this population receptive field mapping that we talked about during the encoding models lecture. This is a rather advanced type of analysis to be performing in a first MRI course, but I've tried to sort of, well, the Dutch word is to pre-chew, but it sounds so gross. So a lot of the analysis has been done for you, but what I hope that does is it gives you a way to step over really the hardest parts of the analysis, but to get to the interesting parts of the analysis. So what this does, if you were to choose this project, you would need to read a couple of papers about how this sort of analysis works and the type of thing that you're going to try to find when you perform this analysis. There's my own little explanation about this here. And then it just dives into, how does this analysis work? Well, quickly, you have a model of what every voxel is doing, right? And that model is that every voxel likes a specific portion of the visual field of the screen, right? And what this means is that we can create a mathematical function, well, just a Python function really, and that is of a two-dimensional Gaussian distribution. So Gauss is an old German dude with a math lump in his head. He was really good, and so he invented the standard normal distribution, which is also called Gaussian. If you want something that is general in terms of statistics, you always use a Gaussian distribution. That's why this model is a Gaussian model. And here's a bunch of different two-dimensional Gaussians in visual space, right? So these are possible instances that this model can generate. Yeah. Here, the cross is the visual field, right? So that's really like what you're looking at. And you can have a model that has, for example, a very small population receptive field at the left top of the screen. You can also have really, really large ones in the right of the screen, and so forth, right? So you have location and coverage, if you will, in this model. And this model can actually find arbitrary shapes like this, yeah? Arbitrarily located and sized shapes like this. Yeah? So how does it do that? Well, that's that's complicated. What it does is it proposes a model, right? So it proposes one of these receptive fields and then says, but I know when the stimulus was on the screen and I know where it was, right? So what I have is actually a movie of the experiment that was run, right? Because I know that I made this experiment. Oh, well, not only did I make this experiment, I am the brain that you're looking at. So I know exactly what was on the screen at each time point. Yeah. So what I can do is say, but if I know this, then I know how strongly this model population receptor field should be activated, right? Because this bar is either in that population receptor field or not. Yeah. And so that's what we're going to base our analysis on. This is when the stimulus is on the screen. It has 4 directions. It's a very simple experiment. This is like the non-Oscar worthiest movie in the world. But for example, what does this lead to, right? So if we have 4 separate model population receptor fields, right? So this one's really close to the center, this one's at the right bottom, this one's at the center top, and this one's right somewhere. What does this mean, right, for what our model thinks the time course will be like? So in a very small, very close to the center of the visual field model, what you will see is that you get these, I think that was this one, the blue one. You don't get very strong activations, but they are very peaked, right? They're not so wide, right? And if you have a wider stimulus, or if you have a wider receptor field model, you will start to get a wider stimulus, or like activations also. Okay? And so what we can do is try to change the location and size of this model to best fit some time course, right? So we can actually generate a lot of different model time courses using this approach. Yeah, any questions? So there, what's on the screen. So what I've done is I've made an image of every TR's screen content. Yeah. So someone's in the experiment. The first, I don't know, 15 TRs. So that's 22.5 seconds. The TR is 1.5 seconds here. You're just looking at the center of the screen. Nothing is on the screen. And then we get this bar appearing at the top and then starting to move down slowly. Yeah. So that's what you're seeing. And of course, then if you have one of these population receptive fields that is at the bottom of the screen, you will get an activation of that PRF's, of that PRF later on during this thing. Whereas if the PRF is at the top of the screen, it would activate early, right? So the time courses that we see below these, you see also that they peak at different times. Yeah. That's because they're placed at different locations, right? So differences in exact timing, the shape and the timing of the signal tell us something about where this PRF likes stimuli to be.
The circles are where it is.
Yes, yes, exactly. So this one would activate early, right? And we can look it up. There's like x0 is minus 0.2, so that's the green one. This one should activate early in the experiment, in the run, because it's at the top, right? And the stimulus began at the top, and voila, right? It actually activates about 4 TRs before any of the others do. So if you had to guess, you can actually do the fitting by hand here, right? You can actually just eyeball it and you know. But of course, we're going to do more than that, right? Sorry? These. Yes, because we know that in the retina, for example, where the information comes in, we have very precise spatial tuning, right? So there, the receptive fields are tiny. They're really, really small. But as we go up in the brain, the higher the visual sort of representations become, the higher the visual processing becomes, the larger the receptive fields become, right? And so we need to have this parameter because this is something that changes in the brain. That's different from region to region, and that's even different within specific regions. Does that clarify things a bit? How does this? Oh, so these are models, right? So like in a GLM analysis, you have a certain expectation of what the bold signal will be looking like. Yeah. Similarly, you can make many different models, right? We've always said you do like a couple of regressors and then you can do T statistics, this and that. Here, you take a different approach and you just try out a whole bunch of models because you're not interested in the statistics, you're interested in which of these models best explains your data. So each of these is a regressor. Each of these time courses is a regressor. Yeah. And then you can just do your standard GLM type analysis. Does that help a bit? So you're not looking at a brain response here, right? You're looking at a model of a brain response. And we want to know which model fits this voxel's data best. And that's the analysis that you'll be performing here. Okay? So you'll be finding for every voxel that has a signal time course, where it likes visual stimuli to be, like what this blob is like for that voxel. Right? So what you'll have is for the entire visual brain, for every voxel, you'll have these blobs that represent where it likes things to be. Yeah. And so what we do is we do what we call a grid search. We make a whole slew of these models and these model predictions, and then we test them all, right? And we just test them one by one, and then we know, okay, this one explains our signal best in this specific voxel. So that means that this specific voxel at this location in the brain likes this portion of the visual field and it has this size. And then we can start doing trickery, right? Then we can start doing things like, well, of course, we need to do convolution with an HRF and that sort of thing. But then we can start to do cool things. Like we find, for example, that this is the blob, the population receptive field that best explains the data from this specific voxel. So you see that you're actually capturing the time course of that response really, really beautifully. And then what you end up is with these values for every vertex. And you can make things like retinotopic maps like so. So if you were to choose this project, you do a lot of this type of fitting and you end up making these maps yourself. Yeah. So you're going to color on the surface of the brain the outcome of this model-based analysis. And then this will give you a way of really inspecting how the visual system processes visual information. Right? So this idea that the visual system is like a convolutional neural network, you'll actually see how that neural network is built up in the brain. Okay? And so there's, for every project, let me highlight this, there is a your task section, and that highlights or outlines exactly what I expect you to do in your report. Yes, this project is kind of a reproduction of the Hubel and Wiesel results, because the movie thing is the bar that they moved around, right? And they recorded from a single neuron. Right? So the difference is we have this predefined movie, it's much slower, and we can look at this receptive field for every vertex in the brain that we have signal for. Yeah? So you end up being able to make really these maps, outline these maps, and analyze these visual representations in detail. So for each of these projects, there are very specified goals that you should strive for, like answers that you should give in your project report. In this case, that, for example, is to replicate certain figures from the original 2008 paper. And then there are subsections to this, which is what they do is they start easy and then they become harder and harder, right? And the final parts are actually hard. So you don't have to finish each and every one of these in your project, right? If you do manage, great, right? But you don't have to. Yeah. And so if you end up being only being able to do up till 2, for example, I don't care, that's fine. That's great. But if you write a good report about that, that does not necessarily mean you get a bad grade at all, right? So it starts to be a bonus thing from about halfway through. Yeah. The better, so the more you can achieve of this, the better it is, of course. But you always need to weigh your ability to also write a report about it, right? So if you end up analyzing this stuff until 5 minutes before the exam, that's not a really wise investment of time. Clear? Yes.
If you do one of the other projects, we have to kind of analyze this as well.
No, but you will be reading the paper. So that is part of the exam material, yeah? And so one of the further extensions, like let's say you're a crack team, like the A team or something, and you're the brain team or whatever, and you finish through even the most advanced parts of these exercises, you can, and they're actually, you can actually start doing another project because at some point they will interface. So you can actually put 2 different sub-projects together if you feel like it. Yeah, you could. So in the Google form that's linked to in the announcement, and I think also in the syllabus, you first make a group and then you decide which project, then you fill it in the Google form. I don't want to give you the finger, sorry. Okay, so that's the first project, right? It sounds a bit daunting at first, but when you read, so the first thing that you do when you start a project is you read the paper jointly, right? Then you talk about the paper with your group. And if you have any questions about the theory at that point, you ask me, right? That's the way to do it. This sort of project, because they're relatively large and they span a couple of weeks, you can get stuck. And so what I want to really say very clearly is don't get stuck, right? Just be structured in the way that you approach the project, right? That is to first read the literature, make sure that you have the concepts clear. And if there's anything that's unclear, ask us. Yeah. But we're here to help, and we're not here to just see you struggle, right? Although we are, of course, incredible sadistic people, that's not our goal. Yeah. The more you can engage with this productively, the more you learn, and that's our goal. So I know, so it says here, right, number 4 and 3 is already advanced bonus material, right? If you want to really go to 4, you read an extra paper, you start doing that. It's not, like in terms of coding, it's not incredibly complex. Oops. I touched this and I shouldn't. There we are. I'm still screen sharing, right? Great. Yeah, so that's clear, right? You can take this as far as you want, right? Okay, good. So I hope that this highlights a bit. I will add this literature to Perusall, so you can actually share your comments on this. Yes, I'll do that today. Very good question. Yeah. Any so if it's not really clear what this project is about, that's not what I'm trying to explain here, right? I'm just trying to explain the steps that this project would take and the ultimate goal and the type of thing that you would be doing, right? So this is really based on encoding models and making a computational model of the of what in this case the visual system is doing. Yeah? So it's really on the computational side of things. If you're interested in computational neuroscience, then this is one of your preferred projects, I suppose. Yeah.
Yeah, still which project need more programming skills than the SEO?
Yeah, so this second one, the somatotopic mapper, needs the least programming skills. Most of the work has been already done for you.
Yeah, I want to know which need more. I don't have much theoretical background.
Yeah, so I think if you can rabbit hole, like you can dive into a deep hole of programming with project 1 and project 3 more easily than with project 2. Yeah. So I'll start with project 2, right? That is a different, very different project. Here, we did not a visual mapper, but a somatotopic mapper. And so what people did here, this is a figure from a paper that we're trying to get published, is, and that by one of our PhD students who was the TA last year, not this year anymore. So, yeah. So what she did is she made these little movies of avatar and of an avatar, like a bald dude, like you see here. This head was shown, and then certain parts of the face were moving. But she would also show like hands, and then certain fingers would be moving, or feet. We also moved feet, right? So what we have is a very simple experiment where about 4 times during a run of 4 minutes or so, people are just in the scanner, they're looking at these avatars and they're like seeing a movement happening, and then they're replicating the movement. Yeah. So they're more or less pretending that they are the avatar, and they're trying to move exactly the way that the avatar is also moving. Yeah. And the nice thing is here, you get incredibly large responses, because a simple experiment like this, where you repeat things multiple times and they're actually pretty long stimulations, you get huge bold responses, and you see that. You really get these blocky time courses where signals are just low and then high. So a very strong experiment. It's therefore also very easy to analyze. But you can already tell that there's a sort of twist here, right? It's not just an on-off experiment. We're actually letting the subject move different body parts. And that's because we're interested in how these body parts are represented in the brain. Yeah. So you may have heard of the homunculus, and I'll talk about that in the neuroscience lecture a bit more next week. But we actually have a very nice and ordered representation of the body in our heads. So what that looks like is, I'm going to go a bit further, but the idea is that we have a body map in our brains, we have a very similar body map for motor actions and for somatosensation. And we're not trying to separate those two, not the somatosensation part and the motor part, because whenever you move, you feel that you're moving, right? And there's no really clear way of getting around that. And so we're not trying to separate those. But what we can do with an experiment like this is actually find this map. Yeah, we can actually find how the body is represented inside the brain. So there is actually a very an infamous paper that found this homunculus in the brain. Maybe I'll show a movie in the neuroscience lecture. So there's a paper from 1937 by Wilder Penfield. He also got the Nobel Prize for this work. They operated on epilepsy patients in a very, nowadays it looks like a very sort of, I don't know, Stone Age type way. So they would just lift the skull from one hemisphere and just, be looking at the brain there. And then what they would do is take an electrode, not to record, but to stimulate, right? So they would stimulate certain parts of the brain. And so what they did in this beautiful landmark paper from the 30s, 1930s, yeah, not 10 years in the future, is that they would really map out what subjects started to move when they would stimulate, right? So they would stimulate a specific portion and the subject would do this, for example, right? And they really mapped out this entire part of the brain, and they found this very ordered representation, this body map along the central sulcus, so in the motor cortex. And so what the cool thing is about this project is you are able to find that same body map. It's beautifully lateralized. You can do all sorts of beautiful things there. And what's easy about this project is that it's a very basic GLM that you run. So you've already done this analysis in the previous notebooks. It's very simple, but you can focus on different levels of detail. So let me just show you what that would look like, right? So here I just made a single, did a single GLM, 4 different events. I think this is the left index finger or something. I don't know. And then, so this is the predicted signal for moving the left index finger. Yeah. And we can run that and then you find some results. This is actually not really showing that much. Is it? No. So I think if you run it yourself, you'll be able to look around a bit. But you can also do a very full GLM. That is, you make regressors for all the different body parts, and then you run the GLM. So not a single regressor, but all of them at the same time. Yeah. And so that then allows you to do these things like making contrasts, like we looked at in the GLM notebook, right? So now you're actually, so what you're doing is you can take everything that you've learned from the GLM notebook and then apply it, but now on the entire brain and not a single voxel. So that's really the case here. It doesn't require that much work. Now this shows you a few examples of the things that you could do. So this is a contrast versus the left hand movement versus the right hand movement. And this is just to show you that the data in this experiment are really, really powerful in that what you see is a very clear patch of this central sulcus activating for the right hand and for the left hand. There are only right-handed subjects in this experiment. And what you then also see is that you don't have just one homunculus that's in this part of the brain. There's another one on the top here. So it's at the top. It's one of those flaps, right? So it's more in the middle of the brain. And hey, actually, there's a blue patch here that's symmetric also, that there's a red patch there. There's another homunculus. Our brains are filled with little people, right? That's also something that's immediately something that you see here, right? You see that the activations are lateralized. You see that the right hand movements, which is high values in this case, are in the left hemisphere, and the left hand's movements are in the right hemisphere. All of that sort of basic brain architecture, this experiment, this project allows you to find. Yeah, so it's really cool in that sense. And that's what you can focus on with this one. Yeah. Any questions? Yeah, humanic, right? I'm presuming that you're talking about the brain surgery experiment. I hope. So then the last project is more geared towards signal analysis, to trying to understand the shape of the bold response and trying to understand the methods that we have to understand the bold response. Yeah. So if you choose this project, at the end, you will have beautiful pictures of the shape of the bold response throughout, in this case, the visual brain. And you'll have a very, very clear insight into really the core of the course, right? That is this bold response idea, how a GLM works, nuisance regression, all of those things we use here for this analysis. So we've talked about this a bit, but you can estimate the shape of the bold response by this finite impulse response type model. This goes into that. And then it talks a bit about nuisances. And then, so there's specific events in which the screen flashed. They are separated in time according to this distribution. This is only a single run, right? So this is a 4-minute experiment or something like that, which we can then start to analyze. We have this entire design matrix that we try to regress onto the brain. And of course, what this gives us is so it's very similar to the somatotopic mapper experiment of Project 2, in that you just run a GLM and you have a whole bunch of regressors for every time point. Yeah. But in this case, all of these regressors stand for our effort to estimate the shape of the bold response. And that's what we will find. Now, where does a flashing screen elicit any response? That's in the visual system in the back of the brain, right? So where the first project actually focuses on exactly these regions to find really like what parts of the visual field do these regions like. This project really focuses on, okay, well, what are these signals that are in there? So what you can do in an experiment like this is now plot the bold response for every time course, for every time point after the event. So you start out with a more or less blank brain. Yeah, so this is just the brain being blank. White means nothing. Red means activation, and blue means deactivation, right? And I told you to be very cynical about when people say activation and deactivation. So what I mean by that is activation is a positive bold response and deactivation is a negative bold response, right? And if you just imagine the shape of the bold response, what you should be seeing is first activation, right? An increase of the bold response, and then you should see this undershoot that we also talked about in the shape of the bold response, right? So you know what to expect. And so I'm making this for every second or so after the event. And the first couple of seconds, you don't really see anything happening. But then after the 4th second or so, you see that we start to see these red patches in the visual system. And they become stronger and stronger. You really see these activations in the visual system. At the same time, you see deactivations, and that's because whenever you see a stimulus somewhere, you don't see a stimulus somewhere else, and you get inhibition as a result. So you see we have this wide activation, and over time, as we go further down, this becomes blue. So you see that you actually see here on the flat map of the brain that the hemodynamic response function first rises and then goes below 0. Okay? So this really focuses on time. I've told you that fMRI isn't good at time, but it's actually pretty good at time. You can do beautiful things. You can also then inspect the precise shape of the bold response very, very specifically for all voxels involved, for all vertices involved. And what this project asks you to do is really go into detail as to why, what is this shape, what does the shape depend on, and all of that sort of thing. Yeah. So this project, I think, is beautiful because it really ties together the time and the space that we are both working with. And you really get a very, very sort of Fingerspitzengefühl sort of thing for what it is that we are looking at when we are looking at bold responses, right? You're really looking at how the blood flows through someone's brain as they're experiencing something. So I think it's really cool in that sense. A lot of this work can also be done just, copying the code from the initial notebooks. So it is not as coding heavy. You can relatively easily avoid going into a rabbit hole of coding with this project too. Yeah, and also, so again, same thing as with the other two projects, right? There's a few bonus questions. So you can stop, right, at any point, because it's all also about, the package that you end up uploading in the assignment. Can we read what you've written, right? Are you able to capture or put into words why this is a cool project, why you chose it, right? Or maybe you can make that up if you were not given your first choice. Yeah, I hope this is clear. It's a bit, the fun thing is I'm throwing you into the deep end, and the not fun thing is I'm throwing you into the deep end. But I hope that there is, there's these little ladders so you can sort of climb out of the water. So I'm here to help. So are the TAs. And, I'm trying to give you some flotation device, namely the notebook. It tries to really explain what you're supposed to be doing and give you a head start. Yes? Other questions? Excellent. We're done.
May I ask a small question, please?
But of course.
Sure, thank you. Could you please summarize very briefly these projects and maybe compare them to each other, if possible?
Yeah, that's actually a very good point. Let me do that. So, so the PRF project, the first project, is really about how does the brain represent space, represent visual space, and it's pretty heavy on the modeling side of things, and you can end up doing more programming. But so in the end result could be that you get very nice visualizations. So that's something that you can look forward to. Second project is easier in terms of modeling, in terms of intricacies of the GLM. It's simpler to implement in terms of programming. And you can create beautiful maps, but it's a bit more difficult than to create beautiful maps, than in the first project. This stays in terms of coding, etc., and methods very close to what you've seen in the notebooks already. And then the third project also still uses that basic GLM underpinnings, but it focuses much more on time. It adds nuisance regression to the mix, and you really, your end result is more like a movie of what's happening in the brain. What I like about it is that you're really looking at this blood flow thing happening inside the brain. If I were you, I would pick all three.
Thank you. Thank you so much.
Oh, very good. Yeah, so Mika just posted like a personal ad in the chat. So if you want to create a group and just find partners, try to do what I said, right? If you have a strong programming and computational background, try to line up with someone who is more conceptual and has more of a neuroscience type background, right? And vice versa. This will give you a much better shot at writing a well-written report in the end. So I'm sort of assuming that you have a WhatsApp group or something as students that you're saying all sorts of ugly things about me in. So I would say that, you know, put your personal ads in there. So try to create groups in that WhatsApp channel thing. Because I'm going to close this meeting and then this chat will disappear, I'm afraid. So you can also do so like in the discussions on Canvas, but I would just do it on WhatsApp because it sounds like it's easiest to do so. Yeah. So the link to the WhatsApp thing is now in the chat, and I promise I will not follow it. I'm going to delete this link from the thing as soon as possible, but you can follow it right now. Yeah, are we done? I'm really looking forward to all the things that you're going to produce.
Yes.
Is there anyone want to group with me? Me and my friends are from computer science program, and we want to do the project one, and we need a psychologist student.
Ah, good.
Yeah, for that, because teacher always involved. So, yes. Yeah.
So I'm going to close the meeting now. If you need the WhatsApp link, maybe you can like contact other students in some other way. Cheers.